My name is Andrzej Tkacz. I'm originally from Poland, but currently I'm talking to you from Faro at the south tip of Portugal. And this is my really short CV. I have a PhD in molecular microbiology from John Innes Center in Norwich. Where I was a biologist, I was running in the lab doing experiments. And then I moved to Oxford, and I was shifting more and more into bioinformatics. Still working in microbiology field. And last year I moved to Portugal, completely different area. And I found a job as a data scientist. I'm doing some metagenomic data. And I learned about sphere principles and sphere data. And this kind of scheme, this arrow, will probably be reflected in this talk of what I'll be talking about. So what I'm trying to do is first to give you some kind of background. If you're a microbiologist, you will know instinctively what I'm talking about. If you're not, I'll try to make it as clear as possible. So what a lab person, when we describe the experiment, sees is this kind of pattern. I don't know if you can see my cursor, my mouse.
Yes, we can.
Okay, thank you. So if we talk about experiment, a lab person will think about, okay, I need to take a strain from -80, find it in the rack, put it on ice, take it to the lab, grow it on some media that we all know in the lab. And then we can split this experiment into something I call simple. Let's say just move the bacteria onto a different media and see how it reacts to it, for example, in motility. We can make some more complicated experiments where we have to adjust OD, spin down the culture, change the media, and let's say see how it grows using a machine like plate reader. We can use commercial kits in order to look at DNA, RNA, or we can push this experiments more, let's say, into do some assays with a host, like I've been doing a lot, which introduce more and more variables into our experiments. However, when we try to get information about these experiments as an outsider, when we read a publication, what we have access to are mostly conclusions from this experiment. We may see a plot with actual data. There may be the measurements, the actual data somewhere in supplementary. They may be to some level with FAIR principles. They may be findable by machine, maybe not. However, a lot of data is either unpublished, lost, or it's hidden somewhere in material methods. What media was used, the time, the machine used, everything should be in material methods, or not. Actually. So okay, a bit of science. Basically, how did I get into this field? How did I get this idea? And this is experiment back from Oxford, unfinished still. And I'll talk. It's just a snapshot of science, 2 slides. We have a bacterium called Rhizobium leguminosarum VC3841. And this bacteria, when put on semi-solid plates, this is what you see here is a picture of a Petri plate from the top. And we let it to swim. It's a semi-solid agar. And it swims very well in glucose, somewhat in pyruvate, but doesn't swim in succinate. Okay, as simple as that. Scientists will say, okay, maybe just this strain doesn't like succinate. However, we do know it lacks succinate. If you follow my cursor, unfortunately, I couldn't change it from white to anything else. If you look at the mean generation time, basically how it grows in liquid culture, it grows absolutely fine in glucose. It grows fine in succinate. There's no even prefer succinate. Basically, growth does not reflect the motility. However, when we look at the pH of the culture, what we see is that in glucose, when it grows in glucose as a carbon source, it acidifies the media. When it grows in succinate, it alkaline, the media. Okay, very clear, quick experiment. If we add more buffering agent, like MOPS in our case, we see an increase in motility. So seems motility is being influenced by pH, which in turn is influenced by the carbon source we're using. So this pattern of motility drop in organic acid as succinate is only visible for some particular strains we tested. We tested many strains, but we only found it in Rhizobium, leguminosarum, VC, and Trifolium, which are some Rhizobium strains are nodulating plants, legumes. That's why they are interesting in for wider scientific community. Okay, and all right, we see a very clear phenotype, and we don't really understand why pH is so important for Rhizobium, but not for other strains. And I remember, reading some literature from 60s. Someone said that Rhizobium doesn't like acidic soils, but if added extra calcium, they seem to tolerate it better. Okay, so I tested that, added, this is in succinate, added some magnesium, no change, added calcium, and bam, they do definitely swim much better. Though we did not observe any clear difference of calcium on the pH. Hence the cause of increasing motility is not per se by rendering pH changes. So subsequently, we identified genes, we did some expression studies, lipidomics, pull downs. However, ultimately, we don't know why Rhizobium is so sensitive to pH and why is it sensitive to calcium level. And I read tons of papers, and at some point I realized that someone somewhere must have found it. It's just I don't have access to this information, and I don't know how to access this information. So how do we actually get scientific information apart from reading papers? So you can ask ChatGPT to make a very nice essay. However, I mean, this is just some stupid example. I ask ChatGPT something very specific that I know the answer to from a paper, and it actually gives you a wrong answer. I mean, it just takes, I'm not entirely sure what it takes, some abstracts of some papers, maybe someone said something on the internet. It actually gives you opposite answer, but that's not the way to actually sort my problem. I cannot ask ChatGPT why calcium is important for Rhizobium. There are some tools that literally let you to push a manuscript into and to harvest information out from the paper, from text of the paper. But if information is somewhere hidden as a plot in supplementary somewhere else, it's we can't get it. So before I actually tell you what I have in my mind, a few examples. So this is a paper from my former lab in Oxford, where my colleagues were looking at role of oxygen in growth of the strain of interest on glucose and succinate. So I thought, okay, maybe there is something that would explain my phenotypes. However, very quickly I understood that okay, it's actually super hard to understand what actually been done. So this is just an example. I try to understand what growth conditions were used. Okay, and we can read that strains were grown in TY, which is a rich media, trypton and yeast, or in universal minimum salts. Okay, which is derived from some other media with some modifications. If you look at this reference 72, you see that media, this AMS now, is derived from yet another paper from 1975 with another modifications. If you go to that very paper, you have the rest of the recipe. But now the recipe is in grams per liter rather than in molarity. So in the end, you can get to this table, but it will cost you a lot of time and a lot of effort. And there will be still some small things like that. Not everything can be autoclaved at the same time. So this is the media composition. The same happens if you actually try to get the data itself. You might have to go to you read something in a text. It refers you to supplementary. You have to find it. It's actually table S1 is the second link. Then you have to find the legend in order to understand which column you actually need to look for the data you want. So I was interested in some gene. Is it important for growth at this condition or not? And this is just an output. This is just an example. Basically, my point is that if we're trying to, even though this data can be considered fair because the raw data is being submitted, every piece of information, I think, is actually available, though it's not available per se for machines. And even for a human, it's still quite confusing. You need to be quite a specialist in order to understand what's what. The last confusing slide, I hope. If we actually want to get some data, we need to look at the figure, try to understand, okay, so we have output from an experiment, but what is the input? What actually the strain, this bacteria was grown in, what media, for how long, what happened? There are hundreds of things that happened to the bacteria before we took this measurement. In order to understand that, we go to material methods, but yeah, it's manuscripts have to be short, so everything is so condensed that at some point we lose the information. What actually happened? It just says, this is just an example of another paper from my previous lab, that the strains were grown again in TY extract or UMS with appropriate carbon and nitrogen sources. So then you have to go to the figure legend and try to make this whole pattern of what actually happened to the strain. So my idea here is to convert this human readable data sets into computer readable data sets. And I propose to make a table. This is just an example of a table. Effectively a massive table that I had a quick go at. This table, what I would like to contain is every row will be one experiment. I would like to know which manuscript it came from. I'm trying to get the cursor. So let's say if that's publication 1, I'll call this experiment 1.1. What I would like to know, not only what was the measurement, but a lot of metadata. Literally everything we can imagine to be collected, I would like to be collected in this table. I would like to know what is this experiment being based on, some previous experiment. How do they connect to each other? Because this kind of information you can get from a paper if you read it very carefully. And for example, if we say growth conditions, 10 millimolar of glucose was added. Okay, but what does it mean? Is it limiting condition? Is it not? There are hundreds of compounds that were used, or maybe tens. What was oxygen concentration? What was nitrogen? What was calcium? Everything we can think about in growth condition, and the same about measurement. This is what we get from from some plot. For example, that strain was the mean generation time is 3 hours 0.7. But is it recorded in a text? Is it taken from a plot? And was there any other phenotype measured at the same time or not? And just for glucose, this table should be expanded to, for example, to say, is it preferred source for the strain or not? How does it get to pyruvate? How is it transported in? How much ATP it gives? I would like to gather all the information we can think about in one big table, which technically cannot be Excel spreadsheet. You will have to be probably some SQL, but also things like antibiotics, what kind of flasks were used, was there any toxicity or at least predicted toxicity of some product, what equipment was used, how long was the measurement, was it the important thing, was it unexpected or problematic phenotype? Does it collide with previously reported phenotypes from, let's say, some other manuscript? And we could also weight this information based on the publication quality. You know, not all the journals we can consider to be equal. So which phenotype we actually believe in more? So in a nutshell, I would like to harvest or convert a brain of a professor of microbiology into a computer. So if you say to a professor of microbiology that 10 millimolar of glucose was used in experiment, a professor would think first, oh, is it enough? Okay, I think, oh, the EPS, extra polysaccharide level layer will be, it's made of sugar, it will be very thick. That's what he or she will think. Oh, how was this glucose imported? Porins through outer membrane, PTS has to be phosphorylated if it goes through inner membrane, and so on, so on. Did they measure oxygen concentration? And why do they use glucose in the first place? Or did someone tell me something that they get a different weird phenotype if they use glucose in some conditions or in some mutant? You know, for a computer, glucose is GLUCOSE effectively, no? And 10 is more than 9 and less than 11. If you ask ChatGPT, it will find everything it knows about glucose, synthesize it for you, and that's it. But what I would like is effectively move all these questions that someone may have, an expert, into an algorithm. So why do I actually want? Why do I actually want to do that? So if you imagine that we have this table, we have all the growth conditions, you know, hundreds of variables, and for a given measurement type, for a given phenotype, we have data. Can we predict phenotype? Let's say, what would be my phenotype if I use this, and that media in that concentration? Another way around, can you tell me what shall I use in the lab in order to get some defined phenotype? Not too much, not too little, just this. So how do I want it to operate? Basically, an expert will have to harvest information from manuscripts, which is time consuming, into computer readable form, which is probably some kind of, you know, table of data. Some things will have to be probably confirmed in the lab again if some phenotype is very problematic. Some things may be obviously missing or obviously wrong. You know, we're dealing with more than 50 years of literature, and that could hopefully lead to discovery of novel processes on bacterial strain phenotype. That's what I'm interested in, but can actually lead to many other things. So basically, who would benefit and how? That's the question. I would like it to be a free to use tool for scientists, basically a phenotype database, to increase our basic knowledge. What was done, what's missing. And I consider that to be the most fair way to store experimental data in future publications, though that would require data storage and so on. Basically, create a tool that scientists would be able to add their data after publication. A lot of biotechnology could, you know, I'm talking about Rhizobium, leguminosarum, but there are thousands other strains. They could ask, okay, how can I get some phenotype out of my strain? Also, I was thinking for publishers and reviewers would simplify their job a lot. You know, if a professor of microbiology reviews a paper, would look at it very different. So not only let's say that in condition A comparing to B, we observe more EPS, but the actual number, the actual weight of EPS would be also scrutinized. For example, if a young postdoc reviews a paper, would not think, oh, is that a lot? Is that not a lot? Does not have enough knowledge in order to compare it to 50 years of literature. While this AI would do that instantly. Oh, this phenotype is extraordinary. I mean, this is unexpected. And I think this kind of data set would be, in general, a good training ground for AI because we have very well-defined inputs and very well-defined outputs. So for now, it's just an idea. Basically, that lives in my head. That needs funding. Hence, I'm talking to you. And I hope maybe you have some suggestions. Maybe you can tell me what to do with that. Maybe just tell me, forget it. It's a fair data project that is based on manual curation of a lot of data, which has to be done by specialists, not by undergrad students. What I've done, I was working. There are more than 100 publications with Rhizobium, leguminosarum, BioVar 3841, BioVar VC 3841, sorry, in each title and abstract in PubMed. And I predict that once it gets going, it will be like 1-2 days in order to harvest information, which is very optimistic because I tried to do 2 papers and at some point I gave up. There was just more, my spreadsheet was just expanding and expanding and expanding in possibilities. I would also need time and effort to create this database, but also to create tools, AI tool, and to confirm some of the phenotypes if they are correct or not. So yeah, that's all I have for you today, and I hope you have questions and suggestions. Thank you for the time.